you that makes me feel happy. It's you that makes me feel right. It's you that makes me feel crazy. It's you, it's you, it's you. It's you that I wanna get close to. I wanna get close to you like your clothes do. Who could ever fool around now? Who they really wanna settle down to? Gotta find you always on my mind. I gotta be thinking up all of my time, all of my time, all my time. There's you, oh, oh, it's you that makes me feel happy. It's you that makes me feel right. It's you that makes me feel crazy. It's you, it's you, it's you. Hey, it's you that is always up on my mind. I gotta be thinking up all the time. Yeah, that's right. You know what makes me feel right, what makes me feel nice. You make me feel twice. Sure, I wanna get close to, just like all your clothes get to do all over you. We said try me young bum, but I wanna make them sing, mama sing that to you. But I wanna make them feel happy. It's you that makes me feel right. It's you that makes me feel crazy. It's you, it's you, it's you. Oh, yeah, it's you, it's you, it's you. Makes me feel happy. It's you that makes me feel right. It's you that makes me feel crazy. It's you, it's you, it's you. All the people you have a right up to the body are you car. But I wanna make me feel happy. Feel the rhythm and the video way they move it. Are you are? But I wanna make me feel right. You. But I wanna make me feel crazy. It's you, it's you, it's you. Oh, hey, it's you, it's you, it's you. Yeah, it's you, it's you, it's you. It's you that makes me feel happy. It's you that makes me feel right. It's you that makes me feel crazy. It's you, it's you, It's you that I wanna get close to. I wanna get close to you like your clothes do. Who could ever fool around now? Who they really wanna settle down to? Gotta when you're always on my mind. I gotta be thinking up all of my time, all my time. There's you, oh, it's you that makes me feel happy. It's you that makes me feel right. It's you that makes me feel crazy. It's you, it's you, it's you. Hey, it's you that is always up on my mind. I gotta be thinking up all the time. Yeah, that's right. You know what makes me feel right, what makes me feel nice. You make me feel twice. Sure, I wanna get close to, just like all your clothes get to do all over you. Who said try me young bum, but I wanna make them sing, mama sing out of you. But I wanna make them feel happy. It's you that makes me feel right. It's you that makes me feel crazy. It's you, it's you, it's you. Oh, yeah, it's you, it's you, it's you. Feel happy. It's you that makes me feel right. It's you that makes me feel crazy. It's you, it's you, it's you. All the people you have a right to do the fun, they are you. But I wanna make me feel happy. Feel the rhythm and the way they move it. Are you? But I wanna make me feel right. You. But I wanna make me feel crazy. It's you, it's you, it's you. Oh, hey, it's you, it's you, it's you. Yeah, it's you, it's you, it's you.
Good evening, everyone. I'm Jack Van Horn from the University of Southern California. And before we begin our program tonight, Peter Bandettini and I would like to say a few words about some of our membership who we have lost in the past year. So I'd like to ask you to please join me, firstly, in remembering the life and the work of our dear friend Bosco Chan, who died unexpectedly in December of 2016. Xiaofeng Chan was born in Beijing, China in 1966 and moved from Hong Kong to the United States when he was still a teenager. He received his bachelor's degree in computer science from the University of Kansas in 1987 and went on to earn his PhD in computer and information science from the University of Minnesota in 1997. His PhD doctoral thesis was entitled Ideal Observer Analyses of Object Recognition. Since then, his neuroimaging work had been dedicated to the exploration of the human visual system to address basic and translational questions pertaining to vision loss, restoration, and rehabilitation. In spite of his focus on original work, his collaborative and collegial spirit was legendary. As such, one needed only to really know him by Bosco. Excuse me. Around the campuses of the University of Southern California, at OHBM meetings and elsewhere, many neuroimaging discussions were peppered with phrases like, "Do you know Bosco? We should inquire with Bosco. Bosco should have seen this," or "I wonder what Bosco's thoughts would be." Indeed, much like Oprah, Madonna, Prince, and others, it was really only his first name alone that was sufficient to represent his intellectual impact, his considerable talent, and his potential for contributions to any neuroimaging project. He was a steadfast teacher, and he served the USC Neuroscience Graduate Program in many capacities, but most importantly in the role of its chair of PhD student selection. Mentoring students was his passion, and he took it very seriously, ensuring that students were welcome to his laboratory, were treated fairly, where he provided critical input on their work and provided thoughtful guidance where it was needed. Recent research of which he was particularly proud of was performing functional MRI studies of visual stimulation in achiasmic patients, that is, patients who are born lacking an intact optic chiasm, as a unique model for studying the visual system. Here, he and his team showed hemisphere-specific retinotopic responses and noted that the amplitude of those responses were proportional to the sum total of the evoked neural response raised to the power of 1/2. This is an elegant study to identify a simple but fundamental power scaling rule of brain physiology. Bosco Chan was a friend, a colleague, an educator, family man, and a dedicated brain scientist. He will be sorely missed. Thank you. Peter?
So this memorial is for Kang Cheng, and so I'd just like to start out by saying that it was a shock to us that Kang Cheng actually died of a heart attack in the December of, or in the fall of last year. So all of us were shocked, and I just wanted to give a few words about what Kang represented to all of us. So to begin, so he was a presence at Riken for a long time, from 2001 to 2016. And he came there, actually it turns out that, so when he came there, this is about 5 years before I met him, he had a 4 Tesla scanner that he, with his colleague Alan Wagoner, worked very hard to extract as much information as possible using that. And he was really pioneering the field. This is probably his, one of his major works. It was one of the first, along with Ravi Menon's work, of looking at ocular dominance columns in humans. And it was actually it was a tour de force. He really put a lot into this. But there was so much more to Kang than just his work, which he was extremely meticulous, he was extremely creative, and he was extremely thoughtful about that. He was always, his science was outstanding, but more importantly, he was a friend. He was a person who was probably the most serene people I knew, and also one of the people who was the most patient as well, and never really had an ego to sort of prove in any sort of sense. And so he made friends with his competitors. There's him inviting Ravi Menon to his lab. He made friends with everyone. I actually, at many meetings around the world, I would always look for him as a person, and I would always try to have dinner with him. I met him first, like I said, about I met him about 1996 or so, and at an MGH when he was taking an fMRI course, and right away he was so approachable, and that's actually what everyone got from him. And I think what's really important to take away from this is that he manifests sort of the essence of a truly international community where you make friends based on the science, you do the good science, you make friends, you have colleagues, and you have relationships across countries. Here's, once again, inviting me to his lab. This is with Nikos Logothidis. This is with Seiji Ogawa, Famid Haider, and his student, Justin Gardner. He was also a great mentor. And this is just an example of how much he loved to interact with students, interact with people who didn't know anything about fMRI. Just basically, he really enjoyed what he did, and you could see it in how he carried himself. And also importantly, he also just recently, and I'm just showing this is actually a paper that's in press as part of a special issue for NeuroImage, and this sort of struck me after the fact because I remember looking at this paper and initially thinking, oh, wouldn't you want to expand it to just the general field? And he's like, no, this very specifically is highlighting my work from when I started to the present date. So I'd recommend taking a look at this paper because it was published, it was accepted about a month before he died. And it is a summary of pretty much his contribution to the field. So he also had a wonderful sense of humor. He was an amazing photographer. He really had a good eye for that sort of thing as well. And he was always taking pictures and always sending and sharing pictures. And I'd like to end with this picture. This is one of many that he's taken of cherry blossoms. Every spring I would look forward to him sending me pictures of cherry blossoms that he would take in Japan. So he will be greatly missed, not only for his science, but for his collegiality and how it represented kind of the best of what our community can generate. The second person I'd like to briefly mention is Andrei Jasmanowicz. So most of you don't know who Andrei Jasmanowicz is. He was a close friend of mine, a colleague of mine at the Medical College of Wisconsin. He was there for over 30 years. He started in the early 80s. And the reason why he's mentioned here at this meeting is that what he did, while it was more technical, it was really helped lay the groundwork of a lot of the pioneering work in fMRI. So he had his PhD from the Nicholas Copernicus University in Torun, Poland in 1975. He was also a singular personality. He was active in the Polish resistance during the early 80s. He actually figured out ways of having radio transmitters self-destruct if they were found. So he was extremely active in that. He left there and just came to the United States and started working in a TV repair shop. And they discovered that he knew a lot more than TV repairs. And then he came to MCW, and the rest is history. He was a driving force in RF coil development for over 30 years. And what people don't really know is also he wrote the predecessor to AFNI. It was called FD, and then FD2. And that actually helped a lot of what we did in terms of our processing in the early days get going. And he was a fountain of ideas. He has 200 abstracts presented at SISMRM, all of them original, all of them his own ideas. And he was completely engaged in the moment, and he had a wonderful sense of humor. He had a wonderful just sense of life. He actually, one of his passions was skydiving. He would film skydiving. He would ride a unicycle to work from his car in the parking lot. He was extremely unorthodox, all in a good way. And one thing that's very important as well is that another example of more than just his science, he was completely engaged in the moment all the time. If I haven't talked to him for over 5 years when I left MCW, he would just call me up and say, I have a question. What about this? And he was never like, oh, let's talk a little bit, let's have some chit-chat. He was lighthearted, but at the same time, completely focused on the moment. And he was a little bit hard to understand. And I always thought it was his Polish accent, but most of his colleagues from Poland said, no, he's still hard to understand in Polish. But because his thoughts were he thought very rapidly, thought very deeply, and he had a hard time getting it out. He died tragically of cancer last year, and on his deathbed, he was still discussing with Jim Hyde, who was at MCW until he retired, ideas for coil development. And if only we could, I think that's the way I think we could all aspire to go in some sense, is that, being completely engaged in the thing at hand right up until the end. So with that, I'd just like to memorialize him as well. Thank you.
Good evening, everybody, and welcome to Puerto Rico. I'm Alan Evans, chair for this year. As you all know, HBM 2017 was to be held in San Juan this year. However, after having negotiated the convention venue contracts, it emerged that the spread of the Zika virus was a potential threat to the health of our members. It cost the organization $250,000 to cancel the contract, but this was the right decision. Fortunately for us, Vancouver was able to step in at short notice to host the 2017 conference. As an adopted Canadian, I'm delighted to see the meeting come to Canada and to such an ideal conference setting. Apart from the vibrant downtown core and excellent convention facilities, nature is so close here, with the mountains and the islands and the ocean right next to us. Yesterday, I was flying over the mountains that overlook Vancouver, and, well, if I were ever to move from Montreal, that's not a hint. We have an exciting program to present this year with an outstanding slate of keynote speakers and a wonderful lecture from Carla Shatz and the Talairach Lecture from Carla Shatz. You'll hear more about that shortly from Program Chair Mike Grecius. Last year has been a busy one for the organization as we take on more activities and leadership roles in the neuroscience firmament. OHBM launched a series of task forces on diversity, multimodality brain mapping and publishing, a scientific advisory board, a communication committee, and collaborations with other entities. You'll hear more about these initiatives at the town hall meeting on Wednesday, so don't miss it. With that said, I would like to pass the conch over to the co-chairs of the local organizing committee for HBM 2017, Lara Boyd and Dori Dude.
Thank you, Alan, and everyone, welcome to Vancouver. We're delighted to actually not have to have liquid sunshine for you, but actually the real thing, and that doesn't always happen. I'm Lara Boyd. I'm from the University of British Columbia. I'm one of your co-chairs here for the local organizing committee. And when Doris and I agreed to host this conference, 2020 sounded like a great time to do it, but we've worked it all out, I think. So before I pass this over to Doris, who's going to tell you a little bit more about Vancouver, I want to acknowledge that we gather here on the traditional, the ancestral, and the unceded territories of the Musqueam, the Squamish, and the Tsleil-Waututh First Nations people. And I think that's important for us all to remember. So Doris is going to tell you, with that, all the great places to eat and, importantly, drink, and maybe hang out a little bit in your free time.
Thank you, Lara. Okay, well, I first want to show you the other members that were able to step up in the local organizing committees. They are from UBC, from SFU, from University of Victoria, and from different institutions around UBC. So, as Lara said, Vancouver is known for walking, especially for biking. There are a lot of bike lanes, I'm sure that you have already seen blocking traffic. There is also a lot of microbreweries. There is a lot of things you can do. You can also inform yourself at Science World, and I think we are going to have Wednesday a very nice venue at Science World. You can go to the Vancouver Aquarium. You can go to the art gallery, the Orpheum Theatre. And this is just to give you an idea of the location. We are here, by the way. The aquarium is around here. For those of you who like to actually walk around, you can actually walk around or bike around the entire Stanley Park venue. You can go visit Gaston. You see that line here, that red line. Unless you are very much interested in getting illegal drugs, I suggest that you be very careful going east, meaning that area of here. Gaston, however, is very safe and is a beautiful, beautiful area to visit. For those who actually like art, here, which is Granville Island, you are going to be able to find a lot of little art shops where you can actually see artists do their work. To give you an idea of how far it is, so we are here, and this is basically, you can go through that area around Vancouver in about 20, 30 minutes, depending on how fast you work. If you are interested in shopping, that street, Robson Street, can do a lot of damage to your credit card. I just want to let you know. Yeltan is actually a very nice little area where there is a lot of little shop and cafe. And as you can see, we are about here. This is Stanley Park, and the area around here is about a 30-minute walk and very easy. For the one who are interested in staying one more day, for example, you can actually, I would suggest you take the bus to actually take you to Grouse Mountain. You can stop by the Capilano Suspension Bridge if you are not sensitive to have a heart attack, and then go end up on Grouse Mountain. You can also take some arbor crosses or go to the UBC Museum of Anthropology, which is actually not that far. And if you have any question, please feel free to find Lara or myself, and even better, the Vancouver Tourism Bureau, which is at the entrance of the convention center, and I'm sure we can answer most of your questions.
Well, good evening and welcome again to Vancouver for the 23rd annual meeting of the Organization for Human Brain Mapping. I'm Mike Grachus from Stanford. I'm this year's program chair, and very happy to give you a quick overview, sort of a whirlwind tour of some of the offerings we have this coming week. So as you know, connectivity is a growing theme at our conference, and I'm just going to give you a little taste for the myriad of options you have this week for both electronic connectivity, scientific connectivity, and also, most importantly, human connectivity. So on these, I guess it's a little shifted, but you'll get the picture. This is sort of the electronic connectivity front. So we have complimentary Wi-Fi. You just connect to OHBM Conference 2017. You don't need a password. It seems like it's working beautifully, which hopefully will last through the week. If you haven't downloaded the mobile app yet, I would strongly encourage you to do so. Even for Luddites like myself, this is an incredibly useful app and basically has all the meeting-related information on it at the touch of a button. There's an internet cafe in the ballroom foyer on level 1. And this year, as in recent years, all the abstracts and poster listings will be available only electronically online at the OHBM meeting website. We have a number of social media options for you, including Twitter, LinkedIn, Facebook, and a really fantastic blog that the Communications Committee has put together for human brain mapping. I'd encourage you to check that out for some really interesting scientific and historical profiles. All right, and then in terms of scientific connectivity and human connectivity, we have a number of enticing events in addition to all the fantastic keynote speakers and what I'm sure will be a very compelling Talairach Lecture from Carla Shatz. So these events listed here are occurring throughout the week. The first is this Open Science SIG. SIG is Special Interest Group. The Open Science SIG Hackathon Collaboration Space. This is on the second floor, meeting room 210, and the WorkSpring foyer there. And this is open to all attendees, even those of you who may not be all that computationally savvy like myself. This is a very welcoming group. They'll be continuing their collaborations started in the pre-meeting hackathon. And there's also a really nice series of demonstrations scheduled throughout the week on some open science tools and software that I'd encourage you to check out. Just outside here in the lobby, the Neuro Bureau again is sponsoring the annual OHBM art exhibit with the theme this year of levels of thought from micro to macro to meta. And including with some available for purchase. Sorry, I meant to mention in terms of the hackathon, the funding for that was made possible by the McGill Center for Integrative Neuroscience, the Wellcome Trust, OpenFMRI, International Neuroinformatics Coordinating Facility, and OHBM as well. And then, importantly, if you're looking for work or if you're looking to hire good postdocs, I'd encourage you to check out the electronic job board at the website listed there. So Monday we'll have the awards for the best paper presentations. The following awards will be announced: the Springer Brain Topography's Editor's Choice Award, the Wiley Human Brain Mapping Editor's Choice Awards, plural, and the Elsevier Neuroimage Best Paper Award. Immediately following that will be the local organizing Committee Symposium, which this year will be on myelin water imaging in human brain, and which looks to be fantastic. Then on Monday, we'll have the first of 2 lunchtime symposia through the week. This one will be a publishing roundtable with Morna Conway leading the charge. She's a journal consultant. You'll also hear from a panel of key journal editors. And the goal here is that you'll have an opportunity to interact with them and dialogue with them and get some feedback, ideally towards helping getting your own publications through along the way. And then importantly, Monday evening, we have the Open Science Block Party. This will be at 3 sort of adjacent venues in the 900 block of Granville Street. And this is sponsored again by the Student Postdoc SIG as well as the Neuro Bureau. And they'll be announcing the winners also of the Brain Art competition at that meeting. So students and postdocs can join at 6. Everybody else is welcome at 7. And immediately following will be the Neuro Bureau Gala at the same location. So be sure to check that out. On Tuesday, thinking more about both scientific and human connectivity here, we'll have our first of 2 poster receptions. This will be at 5 p.m. For Monday and Tuesday posters, numbers 1,000 through 2,223. So this is a nice opportunity to biointerface, which is like a Silicon Valley term for talking to people using your mouth and your eyes and your ears to both transmit and receive information directly from other humans, independent of any electronic devices. And these are nice to fuel these interactions. We have both non-alcoholic and alcoholic drinks, and it's a good opportunity for junior trainees to sort of get put through their paces. I have to say I was at a recent conference where I presented a poster myself and was one of these lonely poster presenters at a basic science conference where nobody came up to my poster. It's a lonely place to be. We've all been there. And I would encourage you, particularly the grizzled, sort of senior gray-haired people, when you see a trainee standing at a poster looking kind of lonely, go up and ask them what their science is about. Similarly, if you're at a poster and your neighbor, and both of you don't have any audience, you can ask your neighbor what their poster is about too. All right. On Wednesday, we'll have another lunchtime symposium. I should mention both of these are sort of brown bag, bring your own lunch symposia. The one on Wednesday is, again, led by the student postdoc SIG, and this will be on mentorship and career development. In particular, they're kind of focusing on this early transition, this challenging transition between, say, postdoc and junior faculty positions and sort of leaping over the abyss that I think many of you feel and see. There are a lot of options out there. They're going to have PIs and also industry leaders looking at both academic and industry options for human brain mapping types. Then, as Alan mentioned, Wednesday evening at 5:15, we'll have our town hall forum. This is a very interactive session, and I can tell you now, as program chair over the last year and attending council meetings, that we take your feedback and your suggestions and your questions very seriously, and this is really your time to be heard. We will also at that meeting be announcing the new program leadership, recently elected, as well as the dates and sites for upcoming meetings. And then everybody, the long-awaited club night is Wednesday night at 8 o'clock. This is at Science World, as Doris has pointed out. Make sure you bring your ticket. It's free to attendees. If you have a guest, those tickets are $50 and you can get them at the registration desk. I was peeking at the website for Science World. This is an incredible venue. It's not your typical science museum. Yes, there's the sort of, the miracle of the Krebs cycle and the oceans and tides and birds of prey, but they also, Vancouver being Vancouver, have an entire exhibition dedicated to the psychology and physiology of tattooing. And so if that weren't enough, we also have a DJ and fantastic food trucks, and it promises to be a fantastic evening at club night. And then Thursday, we have our closing comments and meeting highlights session that starts at 2:45. This year, we're lucky to have Pedro Sosa presenting the meeting highlights for us, nicely stepping in for Carl Zillis this year. We'll also be presenting the People's Choice Award, which, if you look at your mobile app, there's on the main page a voting icon. If you press that, you'll see this sort of instruction. So we're going to be voting, the people are going to be voting on the best abstract of the conference. So take a look at that on your app, and I'll be presenting the winner of that at the closing ceremony. Then Thursday evening, last but certainly not least, is the farewell poster reception. So this is for the Wednesday, Thursday posters, numbers 3,000 and up. And I should say people actually attend this. I was at this last year. Again, there's alcohol to fuel biointerfacing. It's a nice way to say goodbye and also hear a lot of good science. And it's reasonably well attended, so don't think that nobody shows up. Please come and take in some more posters before you go. All right, so as an example of how the council listens to the membership after some concerns last year about $25 t-shirts, at last year's OHBM, we've listened, we heard you, and we've reduced our cost this year to $20. These things are flying off the shelves down at registration desk, just $20, 2 beautiful colors, run, don't walk to get your t-shirt. All right, but it turns out I did a little back of the envelope calculation. T-shirts alone will not actually fund the entire meeting. We'd have to sell, I calculate about 500,000 t-shirts in order to fund the entire meeting, which we can't do. So with that, we'd really like to thank our sponsors for their support of this meeting, in particular our platinum sponsor, Siemens, our gold sponsor, EGI, and our silver sponsor, Philips. I'll talk a little bit about the National Science Foundation's contribution as well on the next slide. And then to kick off the award presentations, I'm going to talk briefly about the travel awards. So this year we had the opportunity to, let me get the data here, award 64 attendees with Merit Travel Awards in the amount of $2,000 each, plus an additional 17 individuals from developing countries who received Merit Travel stipends covering their airfare and hotel for a grand total of $185,000 total in travel stipends. So this is a nice opportunity, a nice thing for the OHBM to be able to provide. The list of all the recipients can be found in the on-site program book, which, by the way, I should have mentioned at the beginning, everything I've talked about so far is discussed in much greater detail in the on-site program. And then, so there's a list of 64 attendees. I'm not going to read them all off or any of them off who won these Merit Abstract Awards, plus 17 travel stipends. But I would actually ask those people who received either a travel award or a stipend award to stand up for a moment. Everybody, come on, so we can see who won these. Up on your feet, people, let's go. I should say these are not, congratulations, these are not easy to come by. There are a lot of people that apply for them, and it's a real honor, so congratulations. And then I wanted to mention again that the National Science Foundation was a big contributor to our travel stipend with a grant that they contributed. If you want more information about NSF, National Science Foundation funding opportunities, they actually have a poster number 5,000, where you can get more information on the NSF and their funding options. All right, I think that covers it for me. It's going to be a wonderful week. It's an incredible place to be. Thanks again to Laura and Doris for putting this together on short notice. And with that, I think I'll turn it back over to our council chair, Alan.
All right. So now we move into the awards phase of the proceedings. The first award this year is the Glass Brain Award. The Glass Brain Award is given in recognition of a lifetime achievement by a lead researcher using or facilitating neuroimaging to discover original and influential findings regarding the organization and function of the human brain. It is my pleasure to present this year's Glass Brain Award to Professor David Van Essen. David is alumni endowed professor of neurobiology in the Department of Neuroscience, Washington University School of Medicine in St. Louis. David received his undergraduate degree from the California Institute of Technology and a PhD from Harvard University. He was a postdoctoral fellow in Boston, Norway, and England before joining the Caltech faculty in 1976. In 1992, he moved to Washington University in St. Louis and has chaired the Department of Anatomy and Neurobiology for 2 decades. Dr. Van Essen is internationally known for his research on the structure, function, connectivity, evolution, and development of cerebral cortex in humans and non-human primates. His tension-based theory of morphogenesis proposes a model for how and why the cortex is folded. His laboratory has developed powerful methods of computerized brain mapping with a particular emphasis on surface-based visualization and analysis of cerebral cortex. His lab recently published a multimodal parcellation of human cerebral cortex that can be identified in individuals as well as group averages. He has been a pioneer in neuroinformatics and data sharing efforts for nearly 2 decades. He has written more than 200 peer-reviewed articles and invited publications. Dr. Van Essen was the founding chair of the OHBM and has been a leader in other major professional societies, including president of the Society for Neuroscience. He served as editor-in-chief of the Journal of Neuroscience and is currently a senior editor for eLife. He was a principal investigator of the Human Connectome Project, a highly successful endeavor to map brain function and connectivity in healthy young adults. He is currently PI on 2 lifespan HCP consortium projects. He is a fellow of the American Association for the Advancement of Science, was recently elected to the National Academy of Sciences, and has received many other awards, including several for teaching excellence. David, congratulations on behalf of the organization. Please step forward to receive our organization's highest recognition and to say a few words.
Thank you, Alan, for that generous introduction. I'm truly thrilled to receive this honor and award from the OHBM, and I greatly appreciate the receiving the accolades from the OHBM membership. So for 50 years, my lab has had a research focus, if you can call it that, on structure, function, evolution, development, and connectivity of the cerebral cortex in humans and non-human primates. There is a unifying theme and methodological mantra underlying this emphasis, and that is the power of surface-based analysis for visualizing and characterizing cerebral cortical organization despite the complexity of cortical folds. So my adventures in cartography started in the 1970s with humble origins, literally using pencil and tracing paper to make manually generated flat maps of monkey cerebral cortex. This was clearly a job for computers, but it took literally 2 decades before several labs, including ours, were able to provide the community with computerized cortical cartography tools and for analysis of neuroimaging data. So in 1995, I had the honor of being a keynote lecturer at the very first human brain mapping meeting in Paris organized by Bernard Mazayer. And that led me down an interesting garden path whose next steps were to be associated with the generation of the bylaws for an entity that would provide for continuity in annual meetings of the human brain mapping. But there was a contentious and somewhat amusing, but at times heated debate at the time. Should this be called an organization or should it be called a society? And I remember actually vividly in that the next year's meeting in Boston with Alan Evans chairing the discussion at the town hall meeting and wearing literally an army helmet in case the crowd got too unruly in this debate. You remember, Alan? So, so as it happened, the decision, as you know, was for OHBM, and I had the good fortune and honor of becoming the first chair of the OHBM Council. It's been a tremendous pleasure to see in the ensuing decades the progress and growth of this society, of this organization, but I wanted to mention that I've been pleased to see it evolving through taking on the trappings of what societies often do. So it essentially has assumed the mantle of the S word, the society, in practice, even not in name. So I'm the lucky recipient of the physical glass brain, and I will resist the temptation to break it into shards to give pieces to all the many people in my lab and in various collaborative efforts over the years who made this award the reality. But I do want to mention a few highlights. The special accolades go to the more than 100 investigators and staff associated with the Human Connectome Project, which, as you heard, is a major endeavor to better understand the organization, function, and connectivity of the human brain. And it's done that through improvements in data acquisition, analysis, and data sharing, and provided the community with a treasure trove of information and discoveries that are leading to deeper insights. I want to mention specifically that my co-PI in this effort, Camille Ugerbil, and also Isa Yakub at Minnesota, the excellent analysis group at Oxford, especially Steve Smith and Tim Behrens, major colleagues at Washington University, Deanna Barch and Dan Marcus, and also key informatics members of my lab, Tim Colson and John Harwell. But especially unique accolades go to an exceptionally talented MD PhD student in my lab, Matt Glasser, who despite his young age, was able to make tremendously broad and deep contributions to the evolution of the HCP-style paradigm for data analysis, acquisition, and sharing. And Matt was also the driving force for the multimodal cortical parcellation that Alan mentioned that I must, in all honesty, say is the most important scientific contribution in my view of my scientific career. So the young adult HCP has moved on and has been supplanted by the two lifespan development and aging projects that Alan alluded to, and I want to also acknowledge the outstanding contributions of that team in that energetic consortium. And last but not least, I'd like to thank Isabel, my wife of 48 years, who's been tremendously supportive and tolerant of my scientific passions and obsessions, and is here to enjoy the occasion tonight. So thank you from the bottom of my heart.
Very, very much merited award. The next award is for education in neuroimaging. And this is in recognition of outstanding contributions to education, training, learning, professional development, and outreach to the field of neuroimaging that support meeting challenges, and in particular, contributions that are in consonance with the goals and purposes of OHBM. This year's Education in Neuroimaging Award goes to Professor Mark Cohen. Mark is a pioneer of functional brain imaging using magnetic resonance imaging. He is professor of psychiatry, neurology, radiology, psychology, biomedical physics, and biomedical engineering at the UCLA Samuel Institute for Neuroscience and Behavior and the Staglin Center for Cognitive Neuroscience. He is also a performing musician. Mark did undergraduate studies at both Massachusetts Institute of Technology and Stanford University, where he received a BSc in human biology. He received his PhD in 1985 at Rockefeller University, where he trained under Victor Wilson, Donald Pfaff, and Susan Schwartz-Giblin. His PhD work was on the pudendal nerve evoked response and its modulation by steroid hormones. In 1985, Mark joined the MRI applications group at Siemens Healthcare, where he began a career in MRI focused originally on education and on technological improvements to reduce scan times. From 1988 to 1990, he directed the applications program at Advanced NMR Systems in Woburn, Massachusetts, a small startup dedicated to the creation of a practical echoplanar imaging instrument. He joined the faculty at Harvard and Mass General Hospital in 1990, where he directed the Hyperscan fast imaging laboratory and the MRI education program until 1993. He has been, since 1993, at UCLA, where he has developed, with John Mazziata, the first dedicated functional MRI imaging center. Mark, please come forward. Pens cap. Thanks, Alan. I'm humbled and honored to be here to receive this award. Everybody knows that our field is intensely multidisciplinary, and to be competent requires knowledge of physics, physiology, statistics, psychology, often engineering, computation, and even graphic arts. Very few people come in with that knowledge, and there's an intense need for education in this field. I've been in this business of teaching for about 32 years, as Alan generously pointed out, and much of my work in education has been to organize courses for what now actually amounts to thousands of students who are eager and even hungry to learn about the details and subtleties of doing good science using imaging. Every one of those courses requires the contribution of first-tier scientists to come in and teach them. And I have brought in people, most of whom receive absolutely no compensation for all their work in teaching. They do it instead for the personal satisfaction of passing on their knowledge to a next generation that will in turn use that to gain more knowledge, and in many cases to improve healthcare and even to have impact on society and culture. Throughout my teaching career, I've been endlessly and deeply impressed by the generosity of these hundreds now of outstanding, talented, and busy scientists who joined me in the teaching mission. Remarkably, especially in the UCLA Semel Neuroimaging Training Program, no more than a handful have said no when they're invited to volunteer to join that teaching team. I'd therefore like to dedicate this award to all of those people who've given so much to so many of you. For the neuroimaging training program in particular, I also want to thank the talented and spirited program officer Stephen Grant at the NIH, who understood that there's a major mission in education to raise the standards of excellence expected of imaging scientists. Finally, I want to thank the organization itself. It says society in here for some reason. The organization itself for its own dedication to education, for the excellent programs that it both sponsors and runs at these meetings. And to those of you who have been in our classrooms or online, in person, your passion to learn always has been inspiring. We're in a vibrant and tremendously challenging field. And as we keep learning, we'll keep making progress together. Thank you. Our next award is the Young Investigator Award. The Young Investigator Award recognizes outstanding contributions made by a young investigator to the field of human brain mapping. Our Young Investigator Award for 2017 goes to Lucina Udin. Lucina is Associate Professor in the Department of Psychology at the University of Miami. After her PhD in Cognitive Neuroscience at UCLA in 2006, Dr. Udin did a postdoctoral fellowship at the Child Study Center at New York University. For several years, she worked as a faculty member in psychiatry and behavioral science at the Stanford University School of Medicine. She moved to the University of Miami in 2014, where she currently directs the Brain Connectivity and Cognition Laboratory. Within a cognitive neuroscience framework, Dr. Udin combines functional connectivity analysis of resting state fMRI data and structural connectivity analysis of DTI data to examine the organization of large-scale brain networks supporting executive function. Importantly, her research focuses on dynamic network interactions underlying cognitive inflexibility in neurodevelopmental disorders such as autism. Dr. Udin is the recipient of an NIMH Behavioral Research Award for Innovative New Scientists, a Brains R01, a NARSAD Young Investigator Grant, and a Slivka Ritvo Award for Innovation in Autism Research. Her 80-plus publications have been in many top journals, including Journal of Neuroscience, Cerebral Cortex, JAMA Psychiatry, Biological Psychiatry, PNAS, Trends in Cognitive Neuroscience, and Nature Reviews Neuroscience. Lucina, please come forward to accept the award.
Okay, thanks very much. This is going to look great on my CV. So I'd like to start off by thanking my research group, the Brain Connectivity and Cognition Lab at the University of Miami. I'm only here today because largely I get to take credit for all the work that they do. The energy and enthusiasm and creativity of this group really astounds me every day, and so thanks, guys. Also, I would like to thank my parents for immigrating from Bangladesh to the United States in 1979 and bringing me with them. Hashtag immigrant excellence. And I would like to thank Brian Morrison, who's in the front row taking pictures to send to my parents, for his constant support and for always making dinner and making sure I eat my vegetables. And also, I'd just like to say, I've been coming to OHBM since 2006. This is my favorite meeting. I really love this organization. And I always come away from it thinking that I've really experienced, the most cutting-edge science and had the best conversations. I love the spirit of open science and international collaboration that everyone brings to this meeting every day. And I always look forward to the meeting, and I leave it feeling really enthusiastic about the future. I'm particularly excited about some new initiatives, such as the Gender and Diversity Task Force. And I'm really looking forward to seeing what that group achieves. And I'm also, as a member of the program committee, I'm really excited to see what a serious a role that gender and balance and all these issues play in the selection of speakers at the meeting every year. And I'd just like to say I'm really looking forward to this year's meeting, and thanks again to the council and everyone for being here.
And our last award today is a new one. It's the Replication Award. The Replication Award is made to the best replication study. This award highlights OHBM's commitment to reproducibility in neuroimaging research and designed to help begin to reshape the incentives towards replication. To tell you a little bit more about this, I would like to welcome Dr. Chris Gorgolewski to the stage to say a few words about the Replication Award.
So, as Alan said, the award aims to highlight not only the best, but also the most important replications in neuroimaging. And this year we received, even though it was the first edition, 22 submissions. And boy, I tell you, they were very diverse. We had studies both from the clinical world as well as basic research, meta-analysis, and even methodological replications. And almost all of them were very, very good. And I don't have the time to, unfortunately, I don't have the time to go through all of them. But under this URL and also in the poster that's going to be displayed next to the registration, you will be able to see the process and all of the submissions. However, there was one submission that really stood out, both in terms of the methods as well as the transparency. And it's really a template for how replications should be done. So on behalf of the Laura and John Arnold's Foundation, it is my pleasure to announce the winner of the 2017 OHBM Replication Award is Dr. Walter Buckle.
Where are you? Here. Put him in the middle. You go next. Thank you.
Wow, this is really great. Thank you. I'm really grateful to be here and accept this award. And I would like to thank the Organization for Human Brain Mapping, as well as the Laura and John Arnold Foundation for setting up this award. I think it's a really great way to incentivize replications. And I hope there will be many more replications in the future. And of course, I'd also like to thank my co-authors, especially Irian Wagenmakers, who has been a great mentor during this period. as well as Beert Forsman, my supervisor. So yeah, thanks.
And now we come to the meat of the proceedings. It is a rare privilege for me to introduce Professor Carla Shatz to deliver this year's Talairach Lecture to open HBM 2017. I vividly remember attending one of her lectures at the MNI about 10 years ago, where she weaved a fascinating story that linked neurodevelopment and neurodegeneration through ApoE4. This experience had a profound impact upon my own lab direction, reassuring me that you could indeed study both ends of the age spectrum simultaneously and not lose your mind. Professor Shatz is SAP Family Provostial Professor of Biology and Neurobiology, David Starr Jordan Director of BioX, Stanford's pioneering interdisciplinary biosciences program. She took a BA in chemistry from Radcliffe in 1969, an MPhil in physiology, University College London, 1971, as a Marshall Scholar. She got a PhD in neurobiology from Harvard Medical School in 1976. Professor Shatz has had a stellar career as a neuroscientist. She's illuminated mechanisms of the dynamic interplay between genes and environment that sculpt our brain circuitry. Her many honors include election to the National Academy of Sciences and the Royal Society of London, the Gruber Neuroscience Prize in 2015, the Kavli Prize in Neuroscience in 2016, and the Champalimaud Award in Vision Research for the discovery of mechanisms that allow experience and neural activity to remodel brain circuits. I can think of few people who better epitomize where neuroscience is going than Carla Shatz, and I am delighted to invite her to take the podium for the 2017 Talairach Lecture.
So Alan, thank you very, very much. I want to especially congratulate all the awardees on their achievements, and also especially David Van Essen. David and I were actually 1 billion years ago graduate students together, and it's wonderful to see him here today. So as Alan mentioned, maybe I'd like to try to answer the question today of what in the world is a developmental neuroscientist doing studying Alzheimer's disease? Well, I mean, an easy answer to this question is actually that I'm getting older and I'm getting worried. But there are other reasons, and I want to take you on that journey in a minute. But one of the things I want to say first, because I think it's a wonderful opportunity, is to just share with you my dream, because I'm going to talk to you today about both synapses and pruning and synaptic pruning. And it just maybe bears a minute to think about the scales, the differences in the scales that we operate on. And my dream is really to, at some day, have a way of non-invasively imaging the human brain at the resolution of synapses, which I think would really be awesome and would settle a lot of arguments, and would also obviously illuminate the mechanisms of many human disorders. So I know there's someone in the audience right now who's going to figure that out. So let's try that again. So here's the thought. The idea here that I want to talk about is if we only understood what are the underlying molecular and cellular mechanisms that regulate these developmental critical periods, could we engage them for brain repair and aging, or even to reverse developmental neuropathology? And for me, actually, what I would love is to be able to speak French without an accent. And obviously, that's a very good example of a developmental critical period that passed me by. So wouldn't it be great if I could just take a pill, reopen up my brain to the kind of plasticity that's present during these early developmental periods, and then learn French again without an accent? That would be just wonderful. So how are we going to do that? Because as far as I know, I haven't heard mice, which are good animal models for trying to understand molecular mechanisms. I haven't heard them speaking French recently. But of course, mice, like other mammals, have many other developmental critical periods. And one of the classic examples, actually there are several classic examples of critical periods in the mammalian visual system, including the mouse. And before I go and talk to you about that, I just want to remind you about the circuits in the mammalian visual system. And those circuits include the connections originating the output neurons of the eye, the retinal ganglion cells, their targets in the lateral geniculate nucleus, and then those targets of those neurons in the primary visual cortex. And the brain has a problem during development, which is it has to actually take two separate images of the world, one from the right eye and one from the left eye, and combine them seamlessly so that we have a perfectly good stereoscopic and binocular view of the world. And of course, part of how this happens is by wiring of circuits. But it turns out the brain also has to learn how to use both eyes together, and that happens during a developmental critical period. So the ganglion cell axons send their connections up to the lateral geniculate nucleus, where they end in a series of alternating right eye layers. And the LGN neurons, in turn, send their connections up to primary visual cortex. And those connections are interdigitated right eye, left eye, or green, pink, as you see here, in this very beautiful pattern that ultimately creates the binocular neurons of the primary visual cortex in such a way that ultimately each binocular neuron receives input from the same part of the visual world in each eye, the right eye and the left eye. This is kind of a miracle. Now, many years ago, David Hubel and Torsten Wiesel, and actually Simon LeVay, who was a postdoc in their lab at the time, worked out a very nice method for labeling the synaptic inputs coming from the lateral geniculate nucleus, from one eye only. And basically what they did was to inject one eye with a tracer substance that goes across the synapse in the LGN, representing that eye, and then is carried yet another synapse up to the terminals in the primary visual cortex. And when they did this experiment, they got this very beautiful, and I hope to you familiar and famous pattern of these ocular dominant stripes in primary visual cortex. And at this level of resolution, every little white dot here actually is about the size of a synapse, in this case, the presynaptic nerve terminal. And the experiment here was imagined to inject a white tracer into the right eye, let's just say. And so you actually can see this wonderful right eye, left eye, right eye, left eye, right eye, left eye pattern of connectivity. Now, years ago, they did a very important experiment because when they first discovered this pattern of connectivity, everybody said it had to be hardwired. It's much too perfect to allow any chance to happen, so it probably is just all hardwired. But they did a key experiment trying to understand the underlying mechanism for congenital cataract. And what I mean by that is, you know perfectly well that if you or your grandmother has a cataract as an adult, having grown up with perfectly normal vision for her entire life, then in fact, she's blind during the time she has the cataract, but as soon as the optics are corrected, she can see again very well, even if she has the cataract for many years. In contrast, if a child has a cataract, let's say a congenital cataract, and that is not corrected soon after, then in fact the child has a difficult time recovering, and if the correction isn't done until after the age of 6 years, then in fact there's a permanent loss of vision in the eye with a cataract. So this is weird. What's the difference between the child who has the cataract and the adult who has the cataract? And the answer, as David Hubel and Torsten Wiesel discovered, is that there's an early postnatal critical period in us. In humans, it's up to about the age of 6 years. There's actually a critical period in the mouse and in the cat and in the monkey and other animal models. And during this time, normal vision through both eyes is essential for wiring up the primary visual cortex in a way that permits each eye to have equal territories in this beautiful right eye, left eye pattern. And instead, if one eye is closed during this critical period, as in this animal model, in fact, this is a monkey visual cortex, and even though if it's just even closed for a few weeks during this time, then the open eye, in fact, expands to take up way more than its fair share of territory. And the closed eye weakens, and the synapses are actually pruned and removed. And so here the open eye was injected with the white tracer, and you can see this white synapses everywhere, basically, except for very small little islands of black in a sea of white. And this reorganization of the connectivity in primary visual cortex is probably still one of our best examples of how experience and use is required for the proper formation of adult circuitry. And in fact, it happens during a critical period in the sense that if one eye is closed in adult in a monkey, for example, even if it's closed and kept closed for a number of years and then reopened, this pattern, once formed, is stable, and therefore there isn't this kind of expansion and so on. And the other point that I want to make here is that, in fact, there's no recovery afterwards, and that's known as amblyopia. In other words, there's a major loss of vision or visual acuity in the eye that's closed. So this is a somewhat almost exclusively irreversible process. So what is going on during these critical periods, and is it actually possible to reverse them? Well, many labs now have discovered that during these critical periods, there's extensive activity-dependent synapse pruning and/or strengthening. And I want to emphasize this. These critical periods are not just about pruning. It's a use it or lose it process. Synapses that are used are actually strengthened, there's growth, and there's stabilization of those connections. On the other hand, synapses that are not used are lost. So there's a kind of relative change in the number of synapses. It's not one thing or the other. And a major question that many of our molecular and cellular labs have now been studying is, how does neural activity drive synapse pruning? And that's what I want to talk about for the next few minutes. So how would you study that? Well, one way we thought of studying this question, trying to address it, is to realize that, in fact, using your brain, so in this case, visual experience, tunes up brain circuits by actually changing the expression of genes that are sensitive to use. So these are activity-dependent genes. So perhaps we can discover genes required for circuit tuning during visual system development simply by conducting a differential expression screen in which we either block activity during development, like let's say block vision, or not, and then compare gene expression in the two conditions, and perhaps we're lucky enough we find some genes that are regulated by activity, and then if we find those genes, then we would have to prove that they're actually required for this activity-dependent pruning. So discover and then prove that they're required. And in fact, now I want to make a bunch of long stories really short, but many years ago we actually conducted such a screen in the presence or absence of activity, and we found a number of candidates, including this particular candidate, very much present in the healthy brain with vision and very much downregulated when neural activity was blocked in the visual system. And here's where we got a huge shock, because what we had discovered was a known major histocompatibility class I gene, a big mouthful, MHC Class I. Now, if you feel weird as if you're sitting in the wrong lecture room now, because MHC Class I genes are the famous genes known to be required for the T cell function and cell-mediated immunity, then you know exactly how we felt when we made this discovery. But I'm here to tell you that not only are some of these MHC Class I genes required for this kind of activity-dependent pruning and remodeling during developmental critical periods in the visual system, both at the LGN level and also at the cortical level, But we believe they're at the synapse, and they may even regulate the composition of postsynaptic glutamate receptors. That's a story for another day. Now, when we made this discovery, let me just say a few more words here. MHC Class I is not one molecule. MHC Class I is a huge gene family. In fact, it's the most polymorphic gene family in our genome. It sits in humans on chromosome 6. There are about 50 of these genes, and it's also true in the mouse. Each one is a transmembrane molecule that, together with beta-2 microglobulin, which is required for stable surface expression, presents peptide to the famous T cell receptor. And when we first made this discovery, of course, our immunology friends got they were very bemused, and more than bemused, they were rather annoyed because MHC Class I proteins were not thought to be expressed in the healthy brain. They're known to be upregulated with damage. But in fact, what I want to tell you is that they're there, and they're actually functioning in important ways at synapses. Now, this was a big surprise. And yet, I want to just now show you some evidence. For example, as I said, there are more than 50 of these MHC Class I genes in the mouse. 3 of them, if we look at the expression pattern of the mRNA, here's a beautiful one that's sitting in layer 6 of visual cortex, including actually even medially and cingulate and so on. Also, a classical MHC Class I gene. These are the ones known to present peptide to the T cell receptor. You can see it here in cortex, in hippocampus, in the thalamus, and so on. And this, by the way, is in the adult brain. And these MHCs change dramatically during development as well. Now, not only is the mRNA there, but the protein is also there. So if you take hippocampal neurons and you put them out in culture and you immunostain for various synaptic markers, here MHC in these little puncta, which are the dendritic spines. We see the MHC expressed in these puncta, and also we see co-localization with synaptic markers. And other labs, including Kim McAllister, have done EM and shown that the protein is actually literally at the synapse. Now, when we first made this discovery, which was many years ago, I went to my computer and I went to PubMed and I typed in, because I thought, what disease might we be studying? And I typed in MHC Class I, Parkinson's, nothing. MHC Class I, autism, nothing. MHC Class I, schizophrenia, an article saying that in a small family study, there was a genetic study, there was an association between the MHC in human, the HLA locus, and schizophrenia. And I thought, that's exciting. I went back to my lab and I told everyone we're studying schizophrenia. And then I made the mistake of a few months later going back and typing in MHC Class I and schizophrenia. And there was another study, more recent, that said lack of association between MHC Class I and schizophrenia. And this went on for years until finally these GWAS studies became hugely powered with thousands of individuals with schizophrenia or controls. And this is an image from a Nature paper in 2014 in which the Manhattan plot of associations basically skyrockets at the MHC HLA locus in humans. And at that time, and even now, there are known to be associations, known HLA alleles and also SNPs at the MHC in the HLA Class I, the classical HLAs, and also some non-classicals that have yet to be pursued, because whenever these papers are published, it's always stated that it's well known that schizophrenia is often accomplished by immune problems as well, and it must be that these HLA molecules are associated with those immune problems. Fair enough, but I'll tell you, they're in the brain. You can go to the Allen Brain Atlas. You can go to a bunch of the other, there are many publications suggesting now that these HLAs are in the human brain, and it's really worth having a different way of thinking about schizophrenia. It's not just the immune system. I'll come back to this. Now, not only that, but last year there was a fantastic paper that came out of the Stanley Center, including work by Beth Stevens. So another molecule that's located in the MHC Class I locus on chromosome 6 is complement, a component of complement. And Beth Stevens and Ben Barris have looked at mouse phenotypes that lack complement. And just to make a long story short, I'm going to say that those are remarkably similar to the phenotypes that we see in mice that lack specific MHC Class I genes. So this is very provocative and exciting. So let me just summarize what I've said so far by reminding you that I'm talking about these MHC Class I genes. We think that they're part of a synaptic system for activity-dependent pruning and plasticity. Perhaps they're altered in schizophrenia, to be determined, and maybe they even collaborate with C1q. Now, MHC Class I molecules have a very short cytoplasmic tail that is not thought to signal, at least in the immune system. We don't take anything for granted, but anyhow, in the immune system, it's known that the way MHC Class I proteins work is to signal, to communicate and signal to target receptors. A good example is the famous T cell receptor. But in the immune system, there are many, many other receptors that belong to the innate immune family of receptors, and these are all known also to bind and signal MHC Class I proteins. And there are many, many of them. They're on B cells, they're on macrophages, they're on some dendritic cells, and so on. And so when we discovered the MHC, we decided to look to see also whether there might be unexpected innate immune receptors also expressed in the brain. And I think I want to tell you about one now, but I'm going to say I think it's actually the tip of the iceberg. So the receptor I want to talk about is called PirB. And let me just explain this receptor. It's a sort of a mouthful, paired immunoglobulin-like receptor B. There's one in mice. There are 5 in humans. They're members of the LILRB family. This is a very wonderful member of the IG superfamily. It has 6 extracellular IG domains, and then a transmembrane domain, and then it has these 5 characteristic ITIMs. These are immunoreceptor tyrosine-based inhibitory motifs. So there are activating innate immune receptors, and there are inhibitory innate immune receptors. And this is an inhibitory receptor, and what that means is that when these ITIMs are phosphorylated, downstream signaling is opposed, including there's a break put on it on MAP kinase signaling and also adhesion signaling cascades. And we thought that was extremely interesting because those are the very cascades that are known to be required or to positively regulate synaptic plasticity. And so we looked in more detail at PirB. It's expressed, this is the protein, it's expressed in the mouse brain. It's very beautifully expressed in the primary visual cortex, one of our favorite parts of the brain, also the hippocampus and other places. So here we have a receptor that's implicated in this whole plasticity story, but is it actually required? And so in order to address this question, we've made a number of different genetic lines of mice, including germline knockouts and also conditional knockouts, where we can knock out PirB gene in cell types of interest and at times and places that we're interested. And so what would we look for in a phenotype in the mouse in terms of ocular dominance plasticity? Well, let me just give you the good news and the bad news. So there are beautiful studies now by quite a number of labs, started by Mike Stryker's lab, demonstrating that mice, too, have a developmental critical period for vision. If you close the eye in a mouse between postnatal day 20 and postnatal day 35, just for a few days, then the open eye actually functionally takes over cortex in the closed eye, in the binocular zone, and the closed eye weakens. And so the whole idea, again, is that the open eye territory expands and the closed eye territory shrinks. Now, the sad news is, to scale, this is the size of the whole mouse visual cortex in relation to monkey visual cortex, and that's not even the whole monkey visual cortex. And Also, I just have to remind you that mice do not have ocular dominance columns, but mice have a binocular zone where the representation of both eyes overlaps. And when you close one eye during these critical periods, the open eye takes over and the closed eye has very few cells left. So you can measure that in a very convenient way. We do it using the induction of an immediate early gene. So we can actually measure, just like in monkey, the expansion in the width of the representation of the open eye in a mouse. So what would we expect in a mouse lacking PirB? Well, in a wild-type mouse that has PirB, the width of the representation here of the ipsilateral eye normally is about 1,000 microns. But if you close one eye during the critical period, it expands. And this is a very nice example of ocular dominance plasticity. So we reasoned that if we knock out the PirB gene and we take an eye out or close an eye during the critical period, there would be no plasticity, no gene, no plasticity. But that's not what we found because we weren't thinking properly. Remember, I told you that signaling downstream of PirB actually acts like a brake on the positive regulators of plasticity. And so what we found, in fact, is that the open eye even takes over more cortical neurons and more cortical territory than in wild type. And this was really unexpected. And not only that, but we also discovered that unlike the adult wild type mouse, where the critical period really closes, in the sense that if you close an eye in an adult mouse that's had normal vision its whole life, the open eye doesn't expand functionally to take over binocular zone. But in these mutant mice, we actually found a little bit of plasticity still remaining in the adult visual cortex. And I got all excited about this because I thought, well, maybe that's enough for me to learn French without an accent. So what's going on in this mouse that somehow allows for more plasticity during the critical period and the persistence of some ocular dominance plasticity even in the adult brain? And actually, you know the answer, and it has to do with synapse pruning, because we reasoned that perhaps there's something different about pruning in these mutant mice, and in fact, maybe there are more synapses that persist into adulthood, and that's exactly what we found. So what we found is that there's a deficiency in pruning. So remember that during normal postnatal development, extensive synaptic remodeling occurs, both pruning and strengthening. And in order to study this process, we bred our mice, either wild type or knockout, with a beautiful line of mice that just marks in their entirety, in a kind of Golgi-like way, layer 5 pyramidal cells, which are a good way to start looking at pruning. So these layer 5 pyramidal cells are marked with green fluorescent protein. They're green. Their axons are marked, and also their dendrites are marked. And if you look at high magnification at these dendrites, you can see these beautiful little protuberances. Now, these are the postsynapses. These are the dendritic spines that receive excitatory synaptic input. So we can count them. These are synapses. We can count them, and that's what Maya did. By the way, Maya's on the job market. And so Maya counted these dendritic spines, and what she found is during the critical period, just at the end of the critical period, when pruning should be finished, there's a persistence of a higher density of dendritic spines, and that also carries on into adulthood, so it's not like a developmental delay. There are just more dendritic spines in germline knockout mice. And not only are there more dendritic spines, these spines may be functional because if we record the synaptic inputs from the layer 5 pyramidal neurons, what we discover is that the frequency of the mini excitatory synaptic inputs is elevated, but there's no change in their amplitude. And this is very consistent with the idea that there are just more excitatory inputs onto these spines. Okay, so at this point, we think there's a role for PirB in not only pruning, but also spine stability. Didn't have time to talk to you about that, but we can do in vivo imaging on cortical pyramidal neurons. Okay, and at this point, this is when our immunology friends said, okay, neurons versus glia. So this is an innate immune receptor. And it's known that actually the macrophages express this receptor. And anyhow, you already studied these MHC Class I molecules, and those are also important in the immune system. So this must all be due to immune function. And then like we go, please, but please, the neurons are expressing these molecules. But I think it's up to us to prove. So we've done quite a number of experiments doing experiments with rescue, where we put things back into the neurons, or selectively removing our favorite molecules, including PirB, just from the neurons. But I want to tell you about one experiment now, which is sort of a favorite of developmental neuroscientists, because the experiment is to make a mosaic using a genetic approach, where one can make a mosaic animal, where the whole animal is wild type. Everything is wild type. The body is wild type. The immune system is wild type. The glia in the brain are wild type. The only things that don't have PirB expressed in them are the neurons. And not just every neuron, but we can make neurons that are isolated in a sea of wild type everything. And then we can ask what happens to the pruning on those neurons. And when that experiment is done, as was done by George Vidal for part of his PhD thesis, George found the same phenotype. That is, in mice in which isolated pyramidal cells lack PirB, those pyramidal cells, both on their apical and their basal dendrites, have more spines than wild type, and also more functional synapses when we record. Now, another thing we can do in this experiment is, instead of looking at postnatal day 30, we can look earlier, during the time just when pruning starts in the mouse. And we can prove that actually pruning is really deficient in these PirB knockout mice by counting the spines about a week earlier. And we do that experiment. What we find out is that both wild type and PirB knockout mice both start out with a high spine density. The spines on the wild type mice are pruned, but the spines on the knockout mice lacking PirB fail to prune. So we really think that this is a pruning deficiency, and we really think that PirB is required for pruning. But what about the glia? Because, in fact, there are beautiful studies from a number of labs, including Ben Barris and Beth Stevens, that have shown that the glia are required for pruning. And let me just say, it takes 2 or maybe even 3 to tango. What I mean by that is the neurons and the microglia and probably also the astrocytes. And how do the astrocytes or the microglia know which synapses to prune? So we think what we're studying here is part of a synaptic tagging mechanism that identifies synapses according to their activity. Remember, we started this whole work by finding genes regulated by neural activity. So we think maybe this is part of the tagging mechanism, and that this is part of how the microglia recognize which synapses ought to be pruned, or part of the protective mechanism which prevents pruning from happening. So let me just summarize again. So what I've told you so far is that PirB, we think, is required for synapse pruning. And let me just say another thing here. So what I think is happening is a kind of a seesaw, and I've drawn it that way on purpose, that there's a balance between pruning and genesis. In fact, maybe there's more pruning than genesis at certain times in life, and more genesis than pruning at other times in life. And what we've discovered is one small component of the mechanism, the molecular mechanism, that is contributing to pruning, that's regulating pruning, and that's PirB. I mean, you could think, what about other molecules that are important for synapse stabilization or genesis? And you could think of TrkB or BDNF or CREB and other well-known molecules. So I think there's going to be a whole set of these pruning molecules, and PirB is one and C1q is another one. Okay, and what I told you is when we knock out PirB, then it's a genetic loss of function, and there's a bias towards synapse stability and synaptogenesis. Okay, so here's where a light bulb went off. Actually, 2 light bulbs went off. And I want to tell you about both of them now, and I'm actually going to kind of start to wrap up, but I especially know that we all would like to go to the reception, and it's beautiful out still, and there's still light out. And I really want to thank everyone who's invited me, by the way, for the honor of giving this talk. So let me tell you about the first light bulb. It's a simple light bulb. You've already thought about it. Here's where the connection is with Alzheimer's disease, because there's excessive synapse pruning in Alzheimer's disease. And in fact, I think many of us now think that pruning is happening well before, patients show up with cognitive loss. Remember, the brain can adapt and is plastic, so the brain can compensate. And certainly, some of this pruning occurs well before neuronal loss, which must happen much later in disease. And let me just remind you that one of the culprits in Alzheimer's disease is a 42 amino acid form of beta amyloid. Normally, that's beta amyloid is produced, or A beta is produced from the precursor protein, the APP protein, by a series of enzymatic cuts and reactions. And normally, these enzymatic reactions produce a large amount of a 40 amino acid form of A beta, of beta amyloid, and a small amount of A beta 42. But if there's a mutation in APP itself in the sequence, or in fact in gamma secretase, which is required for cutting APP and forming the A beta, the 40 amino acid form, then more 42 amino acid beta amyloid is produced. And this 42 amino acid form is capable first of forming oligomers. These oligomers are soluble to begin with, but as they accumulate over time, they actually produce an insoluble plaques, and these are the dreaded plaques that actually can be nowadays detected in non-invasive imaging using contrast agents, and that's part of the diagnosis of Alzheimer's. So one question that we had is, could PirB or the human LILRB be involved in this kind of, in Alzheimer's disease and in driving this excessive pruning? So this would be a gain of function. I talked to you about a loss of function. Now a gain of function. So to address this question, we thought, well, let's just take our PirB mice, the mutant mice that do not have PirB, and cross them to mice that have been humanized, so they become mouse models of human Alzheimer's disease. These mice are transgenic mice, and these particular mice, APP-PS1 transgenic mice, carry a copy of the APP Swedish mutation, human mutation, and also a copy of presenilin 1. It's a deletion. This is the gamma secretase. So it's got two bad hits, this poor mouse. And over time, this mouse, by about 9 months of age, this mouse actually has significant memory loss, can't perform on memory tasks, and also a very big beta amyloid load, and you can see these plaques in the mice. Amazingly, when we looked at these mice with the transgene and the very high beta amyloid load, but without the PirB gene, we discovered that these mice were protected from memory loss, which was really unexpected and exciting. And to try to understand the mechanism, Tae-Ho Kim, who is now at Novartis making drugs, I hope he's going to make a lot of good drugs for all of us, Tae-Ho reasoned maybe PirB actually was binding the APP, this soluble oligomeric form of beta amyloid, and that's exactly what he found. And I'm not going to go into all the gory details, but it's a genuine receptor with a very high affinity binding in the nanomolar range, which is actually even higher affinity than the affinity for the MHC Class I, which is its endogenous ligand. And so now that we know there's a receptor here, that PirB is perhaps being hijacked by the beta amyloid, we can do a lot of experiments, and one kind of experiment we can do is a mass spec experiment where we ask, what are the interacting proteins that are interacting with PirB, both with and without this gain of function, this beta amyloid binding? And again, to make a really long story short, what Tae-Ho found is that among the interacting proteins are two, cofilin, and members of the protein phosphatase 2 family, including calcineurin. And what he noticed is that mice that have a copy of PirB and have the Alzheimer transgene have much less cofulin phosphorylation than mice that, in fact, still have the transgene but lack PirB. In fact, that's sort of a correction. And phosphocofulin regulates the actin cytoskeleton. And when cofulin is dephosphorylated, then it is hyperactivated, and that actually drives actin depolymerization and spine and synapse pruning, which is really interesting because it would be completely consistent with this function of PirB, and now it's been turned on even more. But is this really relevant for human Alzheimer's disease? So, as I said, there's only one PirB in the mouse, but there are 5 human homologs, and these are called leukocyte immunoglobulin-like receptor Bs, 1 through 5. And a number of them are actually, you can find them also in the Allen Brain Atlas. And so we studied 3 of them, which are most closely related to PirB. And by studying which domains of PirB and the LILRBs bind beta amyloid, so we did what's called a domain analysis, we discovered that LILRB2 in these 2 N-terminal most domains, the D1, D2 domain, just like PirB, also bind beta amyloid. And when we looked for LILRB2 in the human brain, in the frontal lobe, we found it was present. So this is a Western blot with an antibody to human LILRB2 from human frontal cortex. So we found it was present, not obviously changed in Alzheimer's disease cortex. But when we looked at the cofulin signaling pathway in the Alzheimer's disease patients, again, we found lower levels of phosphorylated cofulin, again, consistent with the idea that there's more, consistent with the idea that there could be actually a hyperactivation of signaling and actin depolymerization. So it's a correlation, it's not proof of anything, but it's really interesting and tantalizing. And what we think is that it's possible, conceivable, certainly true in the mouse models, but it's conceivable in human that Alzheimer's disease involves pruning, synapse pruning that might be driven by A-beta 42, PirB, or LILRB interactions. Now, if this is so, whoa, so the other light bulb just went off. And that is, the question is there a pill in here? I'm getting to the end. And of course there's a pill in here, and many of you will realize, of course, you could do a large compound high throughput screen looking for drugs that actually interfere with the binding of both the endogenous ligands and also beta amyloid and the PirB protein. You could do that, but there's already a drug, and some of you will know the answer. It's possible to make a decoy recombinant protein. The decoy acts like a sponge. This recombinant protein involves just making the extracellular domain. I mean, it would be really cool just to make the 2 N-terminal most domains. We haven't tried that yet, but proof of principle, we've expressed all 6 Ig domains as a soluble protein. And this thing goes around the brain like a sponge, sopping up all endogenous ligands. And that prevents those endogenous ligands from actually binding to the endogenous receptor and signaling. So it's a loss of function. And this is a very classical way that many famous drug companies, including Genentech and Regeneron, make drugs. These are recombinant decoy receptors. So can we make such a thing? And the answer is yes. We can actually make a soluble PirB mimic. We call it soluble PirB. We can express it. We can make large quantities of it, but it's a protein, so it doesn't get across the blood-brain barrier. But in mice, we're really good at putting proteins into little teeny osmotic mini-pumps and then mini-pumping the protein into the brain. And because this particular protein, we actually designed it with some tags so we could purify the protein and also so we could stain the brain and look to see where it went. We can follow the protein around the brain. So here we've infused soluble PirB. Now what do we do with this protein? Well, we would like to treat Alzheimer's disease, but first we want to just see if it works at all. So what would you do with such a protein? Well, we thought maybe we'd just take adult mice reared with normal vision, infuse this protein, and see if we could reopen the critical period for ocular dominance plasticity. So this is sort of the mouse equivalent of me. Could I take this protein and learn French without an accent as an adult? That's basically it. So that's what we did. We infused the protein in adult mouse brain for about 11 days or so, 10 or 11 days. And then we looked at ocular dominance plus plasticity. And so we know that, because we know that ocular dominance plasticity seems to go hand in hand with elevated spine density, we also looked at layer 5 spine, pyramidal cell spine density. And amazingly to me, the experiment actually worked. So if we infuse in the adult BSA and close one eye or take one eye out in an adult mouse, nothing happens. There's no expansion of the open eye representation. But in the presence of soluble PirB, there's a very significant expansion of the open eye representation, i.e. there's ocular dominance plasticity in the adult mouse brain. But even more amazing to me is that when we actually counted spine density on layer 5 apical dendrites, layer 5 pyramidal neuron apical dendrites, or basal dendrites, we found an elevation in spine density over the control brains. And that's amazing, because what it means is that, within about, a week or so, new spines must have appeared on these dendrites, and we're dying to do in vivo imaging to actually see and watch them and see if they really are appearing. But meanwhile, I can tell you that there's also a change in the number of functional excitatory synaptic inputs onto these neurons, as revealed by making whole cell recordings and recordings of miniature synaptic currents in these neurons. So some of these spines are functional. Okay, so all I can say is yikes, because what we've seen here is that by understanding something about underlying molecular mechanisms of critical periods, we've seen that a receptor that signals, if we block the function of that receptor, we can acutely increase spine density and functional synaptic inputs, even in the adult, mouse brain. But in this case, new synapses seem to be generated. And this is where I really want to sum up now. So this opens up a lot of questions that are fun. One is this a really good approach for treating, pruning disorders? Would you take this as a pill now? How many of you would take this pill right now to learn French as an adult? Very wise, because there's a lot we don't know about the side effects, right? But this is still, I think, rather exciting. The other thing is, I want to just make 2 more comments. First is the brain and the immune system clearly share a common language at the synapse, and by that really I mean molecular language, in the sense that the very same molecules, the innate immune receptor here, and also the MHC Class I proteins that are used by immune cells in the immune system, apparently are also used by neurons at the neuronal synapse. And so there's a way for direct communication to happen, both in the healthy brain, but also with damage and disease. And finally, I wanted to say one other thing. I don't think you can see all these pathways, but these pathways are from a paper in Cell, Expression Analysis of Human Brains from Late Onset Alzheimer's Disease. And again, what's discussed is that there's inflammation in the brain, but I just want to remind you, it's not just the immune system, that these molecules are also being used by neurons, both for normal regulation of synaptic plasticity and pruning, and may be massively dysregulated in disease and possibly in Alzheimer's disease. And so it really opens up many new questions, and I hope that many of you will think about these things next time you read a GWAS study in which the MHC Class I locus is identified as being significant because of immune problems as opposed to changes in brain function of these immune genes. So with that, I really want to thank all of you for the honor of coming to visit in this beautiful place. And also, I know I'm going to learn a lot tomorrow at the meeting itself. And I also really want to thank my lab and all of the funding agencies that have actually taken a big risk on us over the years when we've discovered things that don't fit. And many, many people in the lab who are now doing very well elsewhere, and we couldn't have done any of this without many expert collaborators. It was a true interdisciplinary set of experiments. So thank you again.
So, just a second. So, really, we don't have a tradition of questions for the Talairach Lecture, so I'm sure Carla will be relieved, but we'll be available for questions at the reception. Thank you, Carla, for such a wonderful tour de force, and holding out the prospect of synaptic regeneration in later life is exciting, especially to me.
Yeah, me too.
So it just fails to me to present you with the Talairach Lecture Award. She's the photographer.
She's having good trip.
Okay, but we'll have some photographs. Photographer ran away.
Oh, great. Okay. Thank you. I'll leave this here because.
So that's essentially the end of the proceedings. One of my students, for the first time, has attended this meeting, and I met him this morning at about 11 o'clock. And he said, "I'm exhausted and saturated." I didn't have the heart to tell him he'd got another 5 days of this. Obviously, the best part of this meeting is about to come up now, where we meet our old friends that we haven't seen for the last year. Thank you very much for everybody for coming, and enjoy the meeting.